and sing at the top of my lungs.
Good morning. Good morning. Good morning. Good morning. Welcome. It's Monday. It's it's cloudy. It's snowy again, but this is going to happen. Well done. This is the first unscrambling I've seen. I admire. And it is taken across a gorgeous shot in the Shaw Woods, which is some primary forest in the Ottawa Valley, and of a parasitoid. And parasitoids come into the story today because we are going to talk about diversity. From all of our time together, in 2700 up until today, we've been talking about the number of taxa in the world, the number of species in the world, how this has changed according to different estimates. And we're going to switch today. We're going to unpack where some of those estimates come from, why they're particularly important to a course or to aficionados of invertebrate biology, why it is literally a thing about which we must care a lot. So we're going to leave behind the last chapter, right? Last week we were talking about mobility, and we talked about flying and swimming and walking and crawling and running. And today we're going to run into the next chapter, which is thinking about diversity. And this is the first of those iterations. The second, we're going to talk about potential catastrophic changes to diversity with a specific example of the insects. But today we are going to talk about diversity in general. What does it mean? What is a species? Why is this an invertebrate problem? Elements of discovery and description, differentiating those two things. And then we're going to talk about where some of these estimates that we've tossed around and kind of thrown in or included, where they've come from. We'll talk about Evelyn Hutchinson. We'll talk about Terry Irwin, Bob May, Camilo Mara, Andrew Forbes, and John Wines. And then right at the very end, we'll kind of think about some specific cases or specific areas that we would hope or expect to produce or to preserve diversity. Why are we doing this? What kind of learning outcomes does this tick? What boxes does it tick? What itches does it scratch in this course? Well, it's important. I don't think there's a bigger question, I would argue, in terms of being a biologist. This is our fundamental unit, I will argue to you, of what a species is. So understanding and thinking about what it is, where it is, and why it is are really fundamental aspects to biology. And we're going to unpack and hopefully allow you to become more familiar with thinking about why this is a particularly important question for invertebrate biologists, invertebrate zoologists, or just for the world in general. And I see you. This is not an invertebrate, but it is a very small thing. And since small things are part of the story today, we're going to introduce you to Lily. I don't know if you've seen Lily before. Lily is a miniature dachshund who is, how old are you, Lily? You're 14. Lily the Licker. She is a lovely, yes, you're a lovely dog. And she's passing through. She's going next door into the class next door. Where are we going to start? Let's start out with what is a species? And we're going to start with a very famous biologist and very famous picture of this famous biologist at the end of his life, Charles Darwin, who wrote that no one definition has yet satisfied all naturalists. In his era, a naturalist was a biologist. This was a synonym. But every naturalist knows vaguely what they mean when they speak of a species. And I think this is as true today as it was in Darwin's era. And in fact, you can go through tons and tons of the library, and you can find whole books about the species problem, about the philosophical derivation of the species, what it particularly is. This is the ontology, the ontological truth of what a species is. Or the epistemology, what precisely is a species and how do we measure it? And so all of these books are books that are on my bookshelf, maybe yours, but they're books that are on the bookshelves of your library. And this is a big area of study. And it is called the species problem, or the species problem, because there have been nearly 36 accepted and used definitions of what a species is by biologists. The first one, Ernst Mayr, who lives the longest and so often is the one who dominates discussion and probably who you were introduced to, and I hope not the only definition that you've been introduced to, but certainly the one that you were introduced to first, are groups of potentially or actually interbreeding natural populations that are reproductively isolated from other such groups. Sure, fine. Except hybridization is rampant. And except we never used potentially interbreeding. We never test that. Most taxa of the world, we can't. So we have these other definitions that bring in different aspects of the biology, that bring in different phylogenetic elements of the biology. And so what a species is, often what we end up doing is that bottom one. It's what is the species? It's what a good taxonomist tells you it is. And you say, yes, ma'am, yes, sir, thank you very much. Please tell me when you change your hypothesis so I don't use an out-of-date term or word in the next paper that I publish. And why is this important? Actually, I'll put it to bed. There's a whole course on this that we could take or I could teach. What is a species? A species is a spatio-temporally bounded individual with cohesion mechanisms. That's kind of the answer. How we determine those cohesion mechanisms and how we determine those spatial and temporally those boundaries, that varies from taxon to taxon. And that's why this is more complicated than physics. Physics, a meter is a meter is a meter. It's a fundamental unit. In chemistry, a mole is a mole is a mole. Fundamental unit. In biology, a mole is, well, it depends. It depends when you're asking and what tools you're using to differentiate it. So we have a fundamentally more complicated world than the physicists or the chemists. And this is important because, as Quentin Wheeler wrote, stated bluntly, unless species are known to science and recognizable by scientists, there is no appreciable progress possible in basic or applied biology. Quentin Wheeler is a blunt writer. In fact, he could probably use that prefix in front of most of the sentences that he writes, stated bluntly, ... And this is a problem, and I agree with Quentin Wheeler here. This is a problem because perhaps as few as 1% of the species in the world have names. And this is a problem, or why has this happened? Well, fundamentally, it's about biases, right? It's about biases of some of them that you might be thinking of right now. We would roll into a word that we would call the taxonomic impediment. But perhaps you hear that phrase. Tell me in the chat if you've heard the phrase taxonomic impediment before. A yes or a no, a Y or an N, or if you're as kind of mystified as the 10th Doctor is there. Taxonomic impediment. Oh, good. Amazing. It's a widely used term that basically means anything that slows down an understanding of the species, of the players in the play, of deriving and understanding the fundamental units of biology. So this could be issues currently obstructing taxonomy. It could be the lack of taxonomic expertise. This could be ratios, like the ratio of taxonomists to the diversity that they're studying. And they're just behind, even though they've been working writ large 250 years since Linnaeus. They've been working at understanding these units. They don't understand. Yes, young man, come in. Dougie, come here. Come here. Up. Hi. There we go. There you get 2 dogs, 1 lecture. And so all of these elements, this stagnation, this is important because as one parasitoid taxonomist kind of wrote out in a really interesting figure, this is important because, as the Quentin Wheeler quote suggested, stated bluntly, there's no appreciable progress unless we know what it is we're talking about and we can talk to each other about it. And so the diagram here is kind of 2 ivory towers or 2 castle towers of different branches of biology that study entomology or physiology, or just fill in these word towers on left and right hand side any of the biology that got you interested in going to Guelph and being a biologist. And the orange, the bridges and the bases of those towers is taxonomy, is understanding the units that we work with. So 2 dogs and 1 cat, and I don't see any particular question outstanding, so we keep going. And think about, why is the taxonomic impediment, why is the species problem, why are these particularly invertebrate problems? Well, from a biomass or a diversity perspective, it's because most of the animal mass on the planet are the invertebrates. maybe 3/4 of it, of everything, right? This is a particularly important problem. And one of the players in the story today, we're going to introduce him to you now. This is Robert May talking about the importance of insects in particular as an exemplar taxon of invertebrates.
Insects. More than half the recorded species, roughly 1 million of them, are insects. To a good first approximation, everything's an insect.
I love that quote. We're going to hear more from him today and then the next lecture as well. To a rough approximation, animal life are insects.
Insects.
Right?
More than...
So and then in addition to that, as we discover more and more about the symbiosis, about dependency, about species depending upon one another to live on the planet, we know with increasing certainty that every insect species has likely at least one species-specific species of mite that lives on it, and a species-specific nematode, and potentially species-specific parasitoids. So there are maybe as many as 10. So for every one thing, we've got kind of this diversity upon this accelerant of diversity. So in the most diverse taxon, the insects, we've got all of these fundamental interconnected relationships. Then there's more and more things than we had previously thought. And this has caused some taxonomists, as they think about and make a kind of a journey into the scientific communication world, they wrote about these species landscapes or the speciescapes. This is the first version that came out in the 80s, where the scale of as you looked across at Yosemite or Algonquin or whatever, and the landscape is not dominated by the large bears or lions or tigers or bears, oh my, it's in this case dominated by organisms scaled to their expected or to their named diversity. So when you look out across, you see a diversity of mites and bivalve mussels and beetles. And if we update it today with some numbers that are still out of date, we get a very similar view of the world. So this is not the 80s. This is now in kind of mid-2010s, probably 2015 updating of the speciescape that just emphasizes that what you're looking at on the landscape biology are the diversity of invertebrates by mass or by named taxon. And so if we have a problem with what species are, if we have an impediment to the description of species in taxonomy, these are fundamentally problems of invertebrate biologists. It's a thing that we ought to spend time talking about in a course such as this. And so describing or discovering species, what's the difference? Well, I would say discovering is kind of an older term that you should put away. Don't use it. Nobody discovers a species. A species, it's as wrong or as colonial as thinking about a certain European discovering North America. It was here. The people that lived here knew it was here. These species, we're discovering them only from our perspective where we don't know. So it's a better word to say we're describing new species. And so rates of discovery, so it's kind of I don't know if it's quite a catch-22, but it's a difficult thing because the world in general understands to a greater or lesser degree what discovery means. But description, you need to know a little bit about biology to understand what it is. So it's hard to get people jazzed up at the end of the year to say, wow, these are the newest species described in 2010. And it's like, wah, wah, wah. Instead of the top 10 species discovered in 2025. So they're often used as synonyms, not cinnamon. They are used as synonyms, and I would hope that you go forward in your career using them appropriately to either kind of get interest, but also more accurately to talk about description. And this is really, this is not the death of biology. This is not the death of history. If you've read either of those books or either of those perspectives in a modern era, that's kind of a contemporary bias to think things have happened, most things are done. What we're looking at here is the description of just different groups within decapods and the year that those decapod species were described up until kind of the early 2000s, up until in this graph 2028. And I think it's important to note that about half of all the bird species, birds being our kind of representative of the vertebrates, the well-known things of the world, were described by 1845. And so what fraction of the decapods, of this particular group of crustaceans, as an exemplar of the diversity of the invertebrates, of the diversity of arthropods, were described by 1845, a very small proportion. And then look at the shape of the curve. If we were close to being done describing the players in the play, the ecological and evolutionary dramas about within which we exist, then this curve would go up and would tend to asymptote, would tend to plateau. And we do not see that. And with most groups of invertebrates, we do not see an asymptote. The era of I can talk about this accurately. An era of discovery is what you're living in right now, which is really good news because it's also an era of an obligation to describe things, because there's not just like the birth notices, the species descriptions that we have to do. More often than not, we're writing death notices. We're writing eulogies about species, about taxa that have gone extinct, about which we no longer know where we can no longer find them in the world. And so the rates of description, we'll talk about global or biogeographic biases to that, like equatorial to kind of the polar trends. Lots more species towards the tropics. But our rates of discovery are, and they're biased by that, but they're biased by other things in terms of body size and rough coloration. And that has to do with our biases, the way that we see the world. We've talked about this before a number of times, and I'm bringing it up again. Just the synonym, the bias that's evident in the synonym, meaning when you say I see, meaning I understand, is non-empathetic to most of the taxa that we share, that share their planet with us. These things do not see the world the way that we do. They smell and they taste it, or they see parts of the radiative spectrum that we are completely unaware of. And so this is an example in private backyards in Los Angeles, California. Within a couple of months of sampling them, all of these fly species, gorgeous, right? 36 new species discovered to science, described to science, discovered to the scientists. It was our discovery. They knew they were there, and they were fine interacting with each other. Now, Los Angeles, California, so there's like a geological history, and there's still a latitudinal bias, so it's not a Canadian story, right? Wrong! Here's Canadian examples. These are examples in just areas where I live. This is my backyard 15 years ago, where we found a new species of sawfly to Canada because of a trap we put right in that red box. And this is the woodlot behind where I grew up in the Ottawa Valley, and we put a Malaise trap there, and we documented a new species of parasitoid wasp to Canada from that. There are species being discovered all over the place. In fact, on your Guelph campus, in the Dairy Bush, it has undescribed species. There was a new species of beetle, this one here, of Staphylinid, the rove beetles again from Smith, but Trigodata obscurata, and it was described by Dr. Adam Brunke, who is a former student of this class, who now is a taxonomist of beetles for the Canadian National Collection of Insects, Arachnids, and Nematodes in Ottawa. But the type specimen was collected near Guelph Lake, but I collected it before it was described in the Dairy Bush, in your Dairy Bush. So there are new species around you all the time. There is a latitudinal bias that we'll unpack in a second, but there's also just our bias away from the small smellers and tasters. Most of life is less than a centimeter, weighs less than a gram, and doesn't have a name. And it doesn't have a name because the way we see the world means we don't understand. So it's even the opposite of a synonym. I see means I really don't understand the world for most of the world's biodiversity. Now, in the tropics, I'm going to give you some specific examples of naming where we've gone into. I'm very privileged to work in a protected area in a UNESCO World Heritage Preserve in northwestern Costa Rica called the Área de Conservación Guanacaste. And I've worked there for 20 plus years, and in part of that is me working as a team and working with others around the world to name the diversity there, because, as Wheeler said, unless stated bluntly, unless we have names for things we don't understand, we can't talk to each other about them, and we certainly can't protect them. So this is an outgrowth from a paper that we wrote a few years ago in I think the work with the school children took place in 2015, and I think our paper came out in 2017. With school children, as part of the Programa de Educación Biología, so a biological education program in the park. It works with the communities and the schools within and around the park to inoculate an enthusiasm, or to educate about the diversity of animals in this UNESCO World Heritage Preserve where they live. And so these school children were part of a contest to write a story or draw a picture about the importance of what wild biodiversity means to them. And the winners of that contest, we named species after them. So this is Daniela Lopez, who drew that painting, and this is the species that we named after her about 10 years ago. I love that. I like a lot. Now, I've also worked with this gentleman here. This is Donald Quick, who worked at the Imperial College London, the Natural History Museum in the UK, and now works a lot in Asia, where we have described species of parasitoid. And Donald is a science fiction and a music fan, and so a lot of the species that we described with him were named species that we named after characters from Kill Bill, or species that we named after Lady Gaga. And so the kind of surrealness of patronyms extends to the fact that there are 6 and soon to be 7 species that have been named for me. So that's kind of amazing to me. And so there is a latitudinal bias. There are more things to discover in the tropics, but there are just things to discover amongst the small smellers and tasters that dominate the diversity of the world. And that's at the tips of the tree. Like, this is not done, these are not phyla. And so you might think, Smith, I don't think so maybe there's more species to discover, but the deeper branches of that tree, we kind of understand them. And I have some stories about that as well, because back in 2014, one of the most iconic species discovered that year was Dendrogramma, which are 2 species of Dendrogramma that are pictured here, that at the time was reported, and potentially with great excitement in +1, to be potentially 2 new species from a new phylum that was not recognized historically. Now, when they were collected, they were genetic analyses were not possible, and a couple of years later, when they collected some more samples and they sequenced them, what they found was that the thing they were looking for, Dendrogramma, is right there, and it sits nicely within the cnidarians, and in fact, this is just a little zooid of a siphonophore. So it's a benthic siphonophore, a cnidarian, and what they had discovered was just a chunk of another organism. Not to put too fine a point on it, though, they're at both ends of the tree, from the tips to the trunk. We are still in an era of discovery from our perspective, of understanding the players in this play. Back to Caramel. I don't see any more questions. So now the root of our shared time together. So there's a species problem. There's a taxonomic impediment. We're really kind of behind the 8 ball in terms of we're behind. We've named most birds. We've named most mammals, but we do not have a good understanding, and there's wildly divergent estimates about how many of species are there. And this is important because, as the Mora quote there, knowing the number of species on Earth is one of the most basic and elusive questions in science. Basic because, tying back to Quentin Wheeler's quote, stated bluntly, there's not a lot we can do unless we're sure that we're talking about the same thing. Now, here we go into a bit of the history of science. So this is G. Evelyn Hutchinson, who was a biologist who lived in the first almost entirely through the 20th century. And he has been called by many the ecologist of the 20th century. He wrote a famous paper called The Homage to Santa Rosalia, or Why Are There So Many Kinds of Animals? And in that, he introduced, basically, from first principles, this concept of niche, fundamental and applied niche, and niche partitioning, where species avoid direct competition by adapting to different niches, subtly different niches in the same environment. That separation helps to reduce competition and allows multiple species to exist in the same kind of environment. And so, irrespective of their position in the food chain, he wrote that small animals, and this is one of the important kind of ratios that we come into now, and an idea that I've already unpacked for you. By being small, an animal can become specialized in very small and diversified elements of an environmental mosaic. We're talking about this with our kids this morning, and I was saying that so fundamentally there's like our fundamental niche, our realized niche, it's described by a bunch of things, but there's only so many puzzle holes where this shape fits. But if you are a fraction, like orders of magnitude smaller, there's just more puzzle holes that you could potentially see if you fit there through time. So the degree makes possible a degree of diversity quite unknown among larger groups of organisms. What is he saying there? That just because of body size, we would expect a greater diversity. According to his concept of niche and niche partitioning, there should be a greater diversity of small things. Now, one of Hutchinson's students was Robert MacArthur, who, if Hutchinson wasn't the biologist of the ecologist of the 20th century, then MacArthur was. He lived a tragically short life, and he wrote about the fact that, and he kind of brought some skills in mathematics and modeling to Hutchinson's kind of back of the napkin arguments. And he talked about the relationship between ecological diversity and body size, and kind of formalized what Hutchinson was talking about. So we've already got a mechanism if we think about body size and niches. So there's more niche space, more greater niche partitioning can be achieved by smaller and smaller things. We have a bias away from smaller and smaller things. There should be more smaller things, and we're probably not naming them as quickly as we are the big things. So that's a couple of, Hutchinson and MacArthur. Jump to an entomologist, Terry Irwin, who died early in COVID. And he wrote about this kind of more elegant statement that Wheeler brought up earlier. Only by knowing something's name can you understand it. Can you care about it? Can you work to protect it? And so knowing names gives us our own ecological context. And he's talking really about an interaction. I like that. I called this a more subtle definition because this is him really talking about how he personally, as a human, interacts with other things. So in the 90s or late 80s, Peter Raven, who was a famous, not a topical nominative determinism would have meant that, or kind of guided him towards being an ornithologist, but in fact he is a botanist of one of the biggest botanical collections, the Missouri Botanical Gardens. And Raven wrote Terry in the 80s just to get an estimate, how many species are there in 1 acre of rich tropical forest. Now, Irwin was already using these kinds of insecticides in the canopy to knock down everything that was above, that hitherto impossible to collect. But he would fumigate the canopy and then lay out these sheets on the bottom and collect to get an understanding of all the diversity of insects that were in this kind of vertical stratum above him. And he used, again, a ratio to estimate that for one genus of beetle that he worked with, that he had about 163 host-specific species. Now, the host-specific, this gets him into ratios to say, so where I work, there's this many species of tree. One genus that's pretty well known, and he could speak authoritatively about it, says there's this many species of host-specific beetles in that genus. So then he can kind of scale to say different areas to say, what's the tree diversity in a slightly larger area? Well, if that's the case, and if this ratio of X to Y holds here, does it hold at a bigger area? And then can you use those ratios to work from an acre to the world? And Irwin did. So he thought if 1 hectare forest had 70 tree species, then there ought to be about 11,000 host-specific beetles per hectare. And if beetles are about 40% of all arthropods, then there should be about 31,000 arthropod species per hectare in the tropics. And if the canopy is about 2 times as rich and mostly different species, then he works his way up to get to just under 50, or say just over 40,000 species per hectare in a tropical forest. And that's based on ratios of host specificity. And so then he moves outside of the tropics to say if there's 50,000 tropical trees, then we get to a global estimate that Terry made quite famously of 30 million species of tropical arthropods. This was a much bigger number than had been considered to that point. And remember, we've named we'd named kind of 1.5 to 1.8 at the time million species. So this was saying we were orders of magnitude from being done. Let's bring on a new player. Well, you've seen him already. This is Robert May, who was trained as a physicist, but passionate about biology. So brought his training in mathematics and physics to biology and ecology and applied that kind of big picture thinking to macroecological and epidemiological questions. We lost him at the start of COVID. He's a little bit critical of Irwin's and Hutchinson's estimates, and he thinks they're too crude, essentially. And they build on biases, or they build into biases. So this is him exploring on the left-hand side with his partner and wife, and he talked about the fact that we kind of lose the trend here. So this is a trend based on categories of size, of characteristic body size and characteristic number of species, and we see this kind of a decline, and the slope up. Could we extend this towards the X and Y axis and say what gives us the number of species? And his criticism is in this area here, there be not dragons, but there be tiny things. We're not quite sure if the relationship between diversity and body size extends into the very small things, because this is exactly the groups that we know the least about, biologically, phylogenetically, all of these things. So we tend to overlook, and so yes, this is a crude start. He would have said of Hutchinson and MacArthur's model, but it builds on the things that we know the least about.
One order of magnitude difference, vertebrates to plants in the level of effort. 2 orders of magnitude, vertebrates to invertebrates. No wonder we know so little about them. If this were a business and the accountants were to look at the way it was organized, they would be appalled.
This is from that same lecture where he's talking about the fact that the unit, again with ratios, but the ratio of biologists or taxonomists working on the things that we know, the vertebrates, is enormous. There's many, many ornithologists for every species of bird on the planet. Hundreds of them, maybe thousands, hundreds. There are not enough biologists for the very diverse things. And this is his kind of economy, saying if you were running the business, saying, yeah, we've got all of this inventory, the species on the planet. And by far the majority of it is invertebrate, and by far the minority is vertebrate. But here is the vast diversity of expertise that we have for this tiny fraction of diversity. And here is the 11 people that we have working on all of the cnidarians. And a lot of his role is to catalyze active new areas of research by highlighting biases and assumptions. The biases of extrapolating from better known groups to lesser known groups, when in fact we know so little about those lesser known groups that we don't even know if those same rules applies. And so he advocated for improved sampling. He advocated in tropical areas and a better and just basically that understanding species richness is essential to ecology and evolution.
1 order of magnitude.
I'm going to move on to Camilo Mora. And this is a more contemporary example. Camilo and colleagues worked in, and he used ratios as well, a higher taxon ratio. So not a regional or a region on the globe ratio, but a ratio of within the Linnaean hierarchy: kingdom, phylum, family, order, kingdom, phylum, class, order, family, genus, species. To say, and his proposition was, we probably understand, or we've bumped into those names for the larger categories, we probably have asymptoted. And if we can kind of plot where they've asymptoted and extract that y-axis value, maybe it will tell us where the species, and this is by an assumption that diversity should scale according to this Linnaean hierarchy. Okay. There's something that you're not hearing. I will be right back, and I've got to run and deal with a ongoing question. I'll be right back. There we go. Minor emergency on the first floor avoided. So let's look at this. Instead of looking at the table, yes, I am recording. Let's look at this. So here we've got the number of phylum, through time. So for the category phylum, and we see it tends to asymptote just above 30. Number of class. So we take these asymptote values and we plot them. And so he ends up with, well, where does this suggest we're going to run into an asymptote for the number of species? He comes up with compared to Irwin, a very comparatively conservative number of about 8 million. Now this is my value. I think it's very conservative, but it's an interesting another approach. Instead of a ratio of body size or a ratio of regional diversity, or ratios of diversity one taxon to another based on dependence like host specificity, it's a ratio of Linnaean hierarchy. So they come up with a fairly conservative estimate, but that still says most of the diversity is understudied. And I'd say one of the bigger roles of their paper in PLOS Biology in 2011 was to call attention to the proportion of diversity in the ocean that remains understudied or unknown. Let's talk about the role. We talked about host specificity that was unpacked or kind of brought into the this drama by Terry Irwin, thinking about the host specificity of beetles and trees. And this is where we're going to bring back in parasitoids and bring back in Darwin, who had this famous quote in a letter: "I cannot persuade myself that a beneficent and omnipotent God would have created them," parasitoids. What really would be lost if these abhorred wasps and flies, maybe all flies in general, just vanished. I think if you've ever spent some time in the north, the true north, or even into Algonquin Park in the spring, maybe the back end part of this could be a t-shirt that you could wear that would become blood stained from all of the blood feeding flies. But it does a bit of disservice to the incredibleness that is our parasitoids. And we've talked about them back in the chapters where we were talking about food. We had a whole kind of lecture talking about parasitoids. So just as a quick reminder, we're talking about whether it's eukaryoid or this seed beetle parasitoid here that we filmed in Costa Rica. So inside of that seed, the female is drilling in with her ovipositor because that seed is being eaten by a beetle larva. So that's a hymenopteron that's laying its eggs, inserting it into the seed, the pea seed from the forest floor of the dry forest in Costa Rica, to find the body of the developing coleopteran larva to lay its eggs inside of that. So it's a predator, and every individual kind of predates and kills a single prey item. And all hexapods likely have at least one host-specific parasitoid, and sometimes many, 10 host-specific parasitoids. And then there's a whole thing that we haven't even unpacked that we're not going to get a chance to together about hyperparasitoids. And so those are parasites that would find that seed and know not only which species of seed it is, which species of beetle is in the seed, which species of parasitoid is now in the beetle, and it would then insert its ovipositor to find not the beetle, but to develop inside that wasp. And these are mostly in the insects within dipteran and hymenopteran groups. And when I was a younger, more red-haired, less white-haired biologist with hair, this is the thing that we knew about host specificity in the tropics, is that it shouldn't be. There's so many taxa that you just can't find good examples of host specificity. And in fact, like this species here that we're going to unpack, this is a caterpillar, Rothschildia lebeau, that's eating a specific species of tree. This is a species of tachinid that is looking for the species of tree that is being eaten by that caterpillar. It's going to then lay an egg that's going to be eaten by the caterpillar. That actuates it, and it kind of activates it, begins to develop, and then what we've got is a developing Lepidoptera pupa that has inside it these spiracles, which connect the developing dipteran parasitoid inside to the outside world. And so the indirect method of infection, so the vast diversity of the tropics and the fact that there is sometimes indirect infection, this makes us think that we should see a lot of host generalists. And in fact, for parasitoid biologists for a long time, they would write papers about how there's a mid-latitudinal peak in parasitoid diversity. It's not in the tropics that you see the highest number of parasitoids, they said. It would be in the mid-elevations. Coincidentally, these are the areas where most of the universities of the world exist. It's the backyard for most of the taxonomists. But is it in fact where most of the diversity is? Well, these are a bunch of close-up pictures of a gorgeous tiny wasp that from Costa Rica, that we collected and would have been called, and when we did collect it, it was called Apantales leucostigmus. It's a tiny thing, like a bit of sleep in the corner of your eye. And it was originally described from collections in the Caribbean, in St. Vincent and Grenada, but the range of this species was from Illinois to into the middle of South America. And the story that we told about that was that it was thought to be a generalist predator, and so it could eat many, many different species of developing larval Lepidoptera, and that gave us this wide range for this geographical range for this parasitoid species. What we found nearly 20 years ago when we started sequencing members of this one species is that there are deep genetic divisions within this one species, and they correspond to the different species of host, of caterpillar that they eat. And so in 2008 and then formally in 2014, we took what was one name and we exploded it in one place, just in northwestern Costa Rica, to be 37 different names. So this is a measure of the fact that our estimates, if we're going to use host specificity to kind of as a ratio to say if there's this many host-specific lineages here, and that becomes a bigger area, and this is our global estimate of diversity, this is an accelerant to that ratio. Our work has said there are many more cryptic things than you thought previously. And we wrote that extending these results, even conservatively, would actively set the stage for reconsidering the upper boundaries of the total number of species remaining to be described in the world. This is a colleague, Andrew Forbes, who wrote more about this kind of thing. And he talked about the fact that they're with beetles and with parasitoids. So there's two different, he's written about how host specificity matters for our understanding of global diversity. That in the first scenario, if there's two parasitoids and they all attack 4 species, then the diversity of hosts is greater than the diversity of parasitoids. But if each host only has one parasitoid, then parasitoid diversity outstrips host diversity. This is a beetle example. I'll pull back in some beetles and say, do you remember the staphylinids that we've seen so many pictures of throughout the year? There are already more species of staphylinid beetle, just one family of beetle, than there are all vertebrates combined. And so if there are more parasitoids for each one of those species, then like you see where this is going in terms of unknown diversity and the age of description, or our age of discovery. John Wines is a herpetologist, works a lot with lizards, but he works a lot with understanding cryptic diversity. And he attempted to bring in a review that would look at the effect that molecular estimates of diversity, this kind of technology, the advent of which was in the latter part of the 20th century, but it extends to today. And so they estimated that every morphology-based insect species contains, on average, about 3.1 cryptic species. So our example with the Apantales, that would have been kind of an order of magnitude more than that. That was one name that had 37 species. And they work this ratio forward to come up with an estimate for animals of 100 million, approximately 100 million animal species. And so some of that jump could be seen with different groups. So we have a number of the red tips on the back end or the feet of these examples here are the number of described species of Hymenopteran, of flies, of grasshoppers, of hemipterans, and other kinds of Lepidoptera. Then we have a minimum estimate and a maximum estimate. And you can see in some cases, these are log scales, right? Because the maximum estimate, there's so many species. The world is defined, perhaps biology ought to be defined by the role, the unappreciated role of the things that we don't know, the players in the play that we don't know. Now, finally, last couple of minutes, what I wanted to talk about is just as an example, where are those species, those cryptic species, and where aren't they? So, well, let's deal with the tropics right off the bat. The niche for tropical organisms is often much more finely grained than that for temperate species. And we see greater biotic pressures within and across trophic levels within the tropics, so lots of competition, and that specialization. So the role that MacArthur and that Hutchinson proposed earlier, that has to do with a greater amount, the estimate that there's much more diversity near and around the tropics. And this is likely true, and for many groups it is true. There's some odd taxa, like in a botanical example that comes to mind immediately, willows. The diversity goes the other way. Now, what other areas? Where ought we expect to find a lot of diversity? Mountains. The historic process of mountain building and the abiotic gradients that are immediately there and that occur through mountain erosion and destruction over maybe tens of thousands to millions of years, these result in the concentration in a very small space, particularly when seen from above, particularly in the tropics, very stable abiotic gradients of temperature and precipitation. And this results in mountains across the world, in Canada, in Costa Rica, in Argentina, being either cradles for diversity, for the eruption of new species and taxa, or museums for diversity as areas go through large changes through geological time and maybe perhaps retreat upslope to areas of abiotic, a niche that used to be formerly more widespread. So we ought to see more diversity in the tropics. We ought to see more diversity on mountains. When you have tropical mountains, you see even more diversity. That's where I work in Costa Rica. Now let's talk about another bias, one that we hope, and it has to do again with ratios, is where do we put protected areas? Well, most protected areas of the world are designed with megafauna in mind, the things that we can see with binoculars and the things that we have guidebooks for. But protected areas are not immune to the stressors of the Anthropocene that occur outside of their boundaries. And the insects and other small invertebrates are not, this review would say, adequately protected by the network of protected areas around the world, simply because they're not big enough, they're not diverse enough, they don't take into account. We can't scale from a protected area designed for a large megafauna. It just doesn't capture the diversity of places and spaces that are required to protect the diversity of the biota that might live there, i.e. the very small things. All right. So this is chapter 1. I love this kind of things, but we've talked about a vast variety. We talked about species problems and impediments to taxonomy and ratios used across host specificity, across body size, across the Linnaean hierarchy, across latitude to figure out where the species of the world are. And we have widely divergent estimates from 8 million to 100 million. And we've talked about biases, about where those things exist. Within the biases, one of the common elements that exists is an understanding that the most diverse of the phyla from a species perspective are the arthropods. And the most diverse group within the arthropods are the insects. And what we're going to do next on Wednesday is to talk about one particularly unfortunate, catastrophic thing of the Anthropocene, which is this debate or this observation that insect abundance and diversity is declining. And so we'll talk about that. What can you work on? Keep working on finishing your presentations, your presentation projects. The lecture finals coming up, of course. The case study for this week, again, it'll be available same Thursday to Monday. And I showed this slide last time, but it's just as a reminder that it'll pop up every time that we have together. These are the details as you're preparing yourself for your lecture final. I'm going to stop recording in a second, and I can answer. I see a question that's popped up here. Actually, I'm not going to stop recording because I like this question. Do megafauna tend to need more space? Yes. And the question then extends, wouldn't that help protect smaller things more than protecting the smaller things that would help protect the bigger things? So understand, I'm not proposing that we design exclusively for the small things and extend to the big things, because you're right. I think the latter end of your question is that wouldn't work either. But bigger things also can move better, and they can move farther, and they tend to escape. And so when things go bad, they can escape, and smaller things can't do that. And so even if there's a big space or a space designed for big things, when the feces is hitting the fan, they can move either upslope or across latitude away. Little things can't do that. And so it doesn't capture the processes inherent to the biology of the very small things. All right.